Assuming that we have already uploaded our qPCR data into the application, it is now time to define the experimental conditions of the analysis. These conditions can be defined in two different but compatible ways, either by loading them from an already predefined text file or by creating them from scratch by editing the design panel. In the second option, we need to define every condition of the experiment manually and assign it sample by sample. As we can see, our example consists of 3 brain tissues, amygdala, cerebellum, and a reference group, and each one has been replicated 3 times. In order to avoid repeating this process, the next time we run the analysis, we can save this design into a text file. Since we're using the second option, we have to define our experimental design, assuming we have a tabular text file with our design configuration. Let's reset our design to show how it works. It doesn't matter if it was generated by a former save to file functionality or we already have it, it just needs to follow this format. As we can see, there is one mandatory column named sample name, including the sample names defined in the input files, and a variable optional number of columns where one of them is the experimental factor that we want to use for the analysis. As an exception, plate is an optional column that could be useful when we have 2 samples with the same name on different plates, but this is not the case. Coming back to the experiment design definition, we want to now apply an already predefined design. To do that, we only need to select the file from the experiment design browser, select the experimental factor we want to apply, and check that every factor has been properly loaded. Before applying our design, using the OK button, we have the chance to discard any sample we don't want to include in the analysis. To do that, just uncheck them from the left-hand side of the design panel and they'll be automatically discarded. As we can see, the amygdala group now consists of 3 samples as well as the reference group, and there is no trail of cerebellum samples. To continue this tutorial, let's go back and propose a slightly more complex scenario where we can get into further details of what this option means and the technical group. Although it seems to be the same experiment, in this case the former biological replicates now seem to have exactly the same name. We're now checking the previous example. It is very important to tell StatMiner how to deal with replicates. Does our experiment consist of 3 different tissues and 3 biological replicates, or are we talking about 3 technical replicates of the same sample? By default, StatMiner will treat the same sample name on different plates as technical replicates. For instance, amygdala in plates A, B, and C would be considered the same sample, and therefore replicated detectors will be aggregated. If so, and in our case these are technical replicates, everything is correct and nothing else has to be done. StatMiner will run an analysis consisting of 3 tissues with no biological replicates. On the other hand, if our samples are biological replicates, as in the former example, we would have to give them a unique name at the technical group column. It's very easy to automatically do that by saying to the application that the same sample on different plates is to be treated as a separate sample. StatMiner generates this unique name by prefixing the plate name, and now each sample belongs to a different technical group. In fact, from now on, the analysis will refer to these samples using this name, so biological replicates will be easily differentiated. Just finish by saying that it is possible to add our own technical groups and assign them manually. As expected, samples belonging to the same technical group will be technical replicates and therefore aggregated in the earlier steps of the analysis. So that is all. Our design is done, and now that we have configured the settings, we are ready to perform our analysis. We are going to finish this tutorial by speaking very briefly about the differential expression tools in terms of how to define the new biological conditions that we might want to compare and analyze. Once our main qPCR analysis is done, these tools will allow us to dive deeper into our statistical analysis. For instance, performing a 2-factor analysis, we recommend to check differential expression video to get into details of how to use them. This tool detects the experimental design already created from the wizard, but we also need to define the secondary factor for this variance analysis. To do that, we only need to update current design, adding the new factor, for instance, a time factor. Now the factor conditions, and finally we are going to properly associate each sample with the time condition. That is all we need to add new experimental factor to our analysis. And now, the factor time in this example is available in the ANOVA test as well as in the other statistical tools which we see work in a very similar way. For instance, we are going to open the Lemma tool. And here is the time factor that we've already created in the ANOVA analysis tool. So, we're finished. In this tutorial, we've talked about how to define our experimental factors, how to deal with technical and biological replicates, and finally, how to create new experimental factors that can be used in the context of deep statistical analysis. Next tutorial describes the next step in our analysis, how to configure the settings of the workflow before running the click-and-go automatic analysis.